Clearing the malignancy.
The entire video is going to be on Tregs, also the effects of red cabbage. So the first one is red cabbage downregulates FOXP3 in Tregs, therefore reducing Treg suppression. That blocks TIGIT or TIGIT and TIM3 or TIM3 expression. This is the inhibitory signaling of Tregs. Now Tregs are these cells that cancer cells recruit. Tregs steal IL-2 away from T cells, making the T cells weaker. But the Tregs do something else, which is what red cabbage inhibits. So basically, TIGIT binds to ligands on dendritic cells, CD155. Well, what that does is that it amplifies the suppressive actions and signaling of Tregs, therefore lowering effector T cell activation. So Tregs have many ways of lowering effector T cell activation, including the stealing of IL-2 away from T cells, but that the TIGIT binding 155 on dendritic cells is one way that cancer cells inhibit T cells. So red
the
cabbage
TIGIT-TIM3 pathway is a key immune checkpoint. Cancer cells exploit this pathway to evade immune attack. Now, enter the predator, immune therapy. Red cabbage contains compounds that block TIGIT and TIM3, but Tregs steal IL-2, depriving killer T cells of fuel.
Because the TIGIT plus TIM3 plus Tregs are often highly suppressive, contributing to immune escape. But red cabbage and its compounds blocks TIGIT and TIM3 so that they cannot suppress T cells, therefore restoring T cell activity by preventing overactive Treg suppression. So that consuming red cabbage may weaken TIGIT and TIM3 and Treg suppression, because by suppressing
the TIGIT-TIM3 pathway is a key immune checkpoint. Cancer cells exploit this pathway to evade detection. When TIGIT binds to TIM3, it suppresses immune response. But now, a new predator arrives: immune therapy. Red cabbage contains sulforaphane, which targets cancer metabolism. Meanwhile, Tregs steal
cancer cells. They attack T cells with PD-L1. Cancer cells, that they can even do that with tunneling nanotubes, even from a distance.
Cancer attacks immune cells using tunneling nanotubes, hijacking their power. It hides behind PD-L1, shutting down the immune response. But Predator brings green tea packed with EGC to disable the nanotubes. Green tea activates SCRA5, triggering destruction of cancer's deadly tunnels. Science fights back one molecule at a time.
But red cabbage can modulate PD-1 and PD-L1 pathways. Okay, there's another pathway here called the CCL28 that is on the cancer cell, and the CCR10 pathway, which is on the immune cell. Unfortunately, red cabbage, there are no studies that shows that red cabbage can interfere with this. CCL28 is a chemokine that is produced by tumor cells, especially under low oxygen hypoxic conditions, because cancer cells, they thrive on hypoxia. Now, what could cause hypoxia? Iron deficiency, because the red blood cells are not able to deliver oxygen around the tissues, because red blood cells, they need iron to be able to deliver
iron
oxygen.
Deficiency disrupts oxygen transport.
What could possibly be a
Resveratrol supports magnesium in proper iron
deficiency
metabolism.
could also be magnesium deficiency.
Without balance, hypoxia threatens cellular energy.
Because in lysosomal membrane peroxidation, magnesium becomes deficient if the fire is not put out. Magnesium is supposed to repair the membrane if the lysosome is peroxidized, because if it's not repaired, then the iron is not properly metabolized, therefore leading to a low oxygen or hypoxia because of that iron deficiency. Okay, so basically, Tregs are recruited.
Tumors secrete CCL28 to recruit Tregs via CCR10, but then Tregs steal IL-2, starving effector T cells.
By the CCL28.
Without IL-2, T cells can't fight.
So
the tumor wins.
CCL28 that the cancer cell or tumor cell secretes, it ends up binding onto a receptor on the immune cell called the CCR10. And by doing so, that's how Tregs are recruited. So unfortunately, red cabbage does not or block that mechanism. Red cabbage does not seem to block CCL28 secreted by the cancer cell to bind onto the CCR10 on the immune cell. Some further research on that and cover that in a different video though. Okay, now red cabbage can optimize the environment for CD91 mediated phagocytosis by macrophages. What CD91 is it's actually a classical receptor ligand on the macrophage. Now, what CD91, but in order for that to be able to happen, the KEAP1 or KEAP1 has to let go of the NRF2 because the KEAP1 is antagonistic on the NRF2 or even binds or even degrades the NRF2. Therefore, NRF2
the blocker.
Activation
it binds NRF2, preventing its activation. But NRF2 is needed to boost Calreticulin. Enter Moringa, the key. With Moringa, NRF2 breaks free, boosting Calreticulin for phagocytosis.
Moringa can antagonize or degrade KEAP1, so that KEAP1 releases NRF2, so that NRF2 can enhance phagocytosis. So basically, the CD91 is on the macrophage. Okay, so basically, a CD91 receptor on the macrophage can bind to or act on to the calreticulin or CRT, which is exposed on stressed apoptotic or cancerous cells. So basically, that's how apoptosis happens. The CD91 on the macrophage binds onto the CRT on the cancer cell, but the cancer cell might try to hide it. So vitamin C in red cabbage supports the proper folding and secretion of CRT. In other words, the vitamin C can weaken cancer cells or force cancer cells to secrete or to show their CRT because cancer cells, they try to hide it. The cancer cell tries to hide the CRT. So the vitamin C forces the cancer cell to expose it on the surface.
Let's boost the immune system.
If the CRT is hidden or not exposed on the surface, then the macrophage cannot do phagocytosis with its CD91. So vitamin C combined with Moringa powder is a very lethal effect against cancer cells. Upregulation of CD91 expression by polyphenols promotes anti-inflammatory macrophage phenotypes, M1, M2 transition. I have a very important video on M1, M2 phenotypes, so check that one out. Okay, so a lethal combination is a vitamin C to force out the calreticulin on the cancer cell, to enable the CD91, the KEAP1 has to stop degrading or stop holding on to the NRF2. If the KEAP1 keeps on blocking the NRF2, then the NRF2 cannot support the CD91 on the macrophage. So Moringa will stop KEAP1 from stopping NRF2. This is really important to put into action everything that I already mentioned in this video, because red cabbage can also influence the shift from M1 to M2 phenotype. So basically, M2 phenotype can support tumors because M1 phenotype is anti-tumor. But the polyphenols in red cabbage does that to protect the over-inflammatory activation of an M1 phenotype, because an M1 phenotype actually makes T cells overactive and then starts attacking tissues. So that's why it has to shift from M1 to M2 phenotype, which is repair mode, which tumors exploit to survive. But in order to mitigate that and to protect
clearing the malignancy
and the negative consequences from going to M2 phenotype, the things that were explained from the start of the video can actually help to mitigate or to reduce the severity of the shift from an M1 phenotype to an M2 phenotype. So like, comment, share, subscribe. Leave your comments down below about your own insights and knowledge, and I'll see you in the next video.